is Yang, and I'm your instructor of the week and your tour guide at the interface of engineering and life science. I really appreciate Professor Rigetti's introduction, so I think I want to say just one or two sentences, a little bit longer than that, about how I end up working on what I work on now. Not many years ago, I was like you, and I was engineering student. My research was involving a lot chips from Texas Instruments, and making hardwares, detecting smoke, and taking actions, and so on, signals and systems on hardware. But I always know that I have some curiosity about biology and some desire to do something about disease. It just wasn't clear to me how. Because back then, to me, biology was about experiments to make observations, but I liked more physics, math, programming, and so on. Until graduate studies, I realized that actually all these interests can be all addressed at the interface, because there are a lot of grand challenges that cannot be solved by a single discipline. So there are a lot of needs for people like you with composite education and training background. So it's really a blessing. So a lot of you are class of 2027, right? 2027, yes. 2028? Wait, most are 2029? Class of 2026. Wait, you're graduating this year, most of you? Okay, well, congratulations beforehand. So there are a lot of interest, a lot of diversity in your interest. Okay, some of you are in engineering, you wonder how I can apply what I learned to a new area like biology, right? Some of you might be like me in those years who are always curious about biology and wonder, how do I get into that? And some of you might be, as you are getting close to graduation, might be even exploring career opportunities in this area. So I hope I can share some knowledge with you and happy to answer any questions at any point. Okay? So we are in week 4 now, and we're about to step to the next module, right, the week afterwards. Okay. Okay, so let's get started. So the big idea of the talk here is the following. I just summarized everything here. Proteins are biological parts in living systems. So this module, we actually zoom into the very basic macromolecular level. And we are focusing on a workhorse molecule of life called proteins. And their functions actually depends on their three-dimensional shape or structure. And we are trying to solve two representative engineering problems in this lecture. We're focused more on the first one, that is predicting the three-dimensional structure. So input is a sequence and output is a structure. And the second one is designing new proteins. And it's sort of the inverse problem to structure prediction. The input is a structure or even a function, and the output is a protein sequence. Okay, we'll explain what they mean later on. And AI actually accelerated both, and this connects directly to drug discovery, and we'll talk more about that in the next lecture. But in the next lecture, we're not just talking about AI for drug discovery, but we'll also give you a broader idea about how AI impacts healthcare in general. Okay, so that's the big idea. So the promise of the day is you will be able to explain why structure or the shape is a bridge between the protein sequence that defines the protein and the protein function that tells what it does. Okay? And you will be able to distinguish these two types of problems, prediction and design, as forward versus inverse problems. And lastly, you will be able to at least name one way AI changed structure prediction or protein design, okay, at headline level. So that's a promise. How do we get there? So the roadmap of the day is following, okay? We'll start with the explanation or introduction of proteins as a basic part of biological system. And we'll explain why its structure or shape matters. And then present a gap for such structure, which motivate the prediction problem starting from sequence and ending up with structure prediction. Okay? So that's a majority of the lecture today. And then we talk briefly about the inverse design problem, starting with the desired structure or function, and then end up with the sequence design. At the end, we'll briefly talk about the AI wave, because we'll explain both the classical and the new formulation and algorithms. Then gradually we'll show the connection to drug discovery, which will be covered in the next lecture. But again, in the next lecture, when we focus more on AI and deep learning, we'll start with broader impact, not just on drug discovery, but on healthcare in more aspects. Okay? So let's get started. We are familiar with engineering systems, and we are very familiar with the parts like resistors, capacitors, inductors that make logic gates, that make modules, that make computers, that make high-performance computing center. This is a very familiar concept to us. And one characteristic of engineering system is it's hierarchical, apparently. And this characteristic is shared with natural or biological systems. So at the part level, there's no resistor, capacitor, or inductor in our bodies for clearly, but there are DNAs, RNAs, and proteins. There are no wiring in our body, but these proteins, DNAs, and RNAs interact with each other, react with each other. They form pathways that form cells, tissues, organs, humans, and human societies. So this hierarchy is shared between engineering and the biological system. That's why understanding and engineering biological systems is not a foreign subject to engineering. It is our subject. In fact, before you and I were born, in 1948, this is Norbert Wiener. You have learned signals and systems, right? And you haven't learned Wiener filter, but you probably have learned Kalman filter. But anyway, this is Norbert Wiener, who published a book in 1948. It's called Cybernetics. Cybernetics was a name for the AI before the word AI was born. But look at the subtitle. Control and communication in the animal and the machine. What does it say? It says that he, back then, already recognized the similarity between the natural and the engineering systems, right? Clearly, there are all kinds of signals in such systems. What are signals? Functions that carry information, right? But there are all kinds of signals, acoustic, vision, or biological, right? Brain or sensor, all kinds of signals. And these signals are communicating together, shaping life. They can be sensed, they could be understood, and they can be modulated as needed, for instance, to treat diseases. So, so this is why I think the understanding and modulating biological systems is part of our engineering quest as well. So what parts are there? I said before, there's no resistors, capacitors, and inductors. Instead, there are three typical macromolecules, DNA, RNA, and proteins, that carry and relay and transform the information in our body. That's really amazing. What information? Well, to start with, there's genetic information, which is in the form of a DNA sequence, right? So that genetic information can be copied from DNA to DNA. So that's why there's a resemblance between siblings or between parents and children, right? And that genetic information in DNA is actually kept in a very safe place of the cell, specifically within the nucleus, with controlled access. Who can access that confidential information? Well, there's a molecule called messenger RNA. The M in front of it means it's a messenger. There are all kinds of RNAs. This is a messenger. Just as the name suggests, messenger RNA will actually transfer that information. It will transcribe that information as if it's copying the data from the computer to a hard disk, to a jump drive. And then this information, right, now can be taken outside of the nucleus into the cytoplasm of the cell, where proteins can be assembled accordingly. So that genetic information, is instructing what proteins should be made. And that process is called translation, okay? So why transcription for DNA to RNA information flow, but translation for RNA to protein information flow? Because DNA and RNAs are similar, they're of similar building blocks called nucleotides. So it's like, transcription, copying data from one media to another medium. But RNAs and proteins are in different language. Proteins are in the language of amino acids, unlike RNAs in the language of nucleotides. So that information change is called translation. So this is just the fundamental of the information flowing across the parts, and there are a lot of things we still don't understand. There's a 3-minute video if you are interested in what we do know about this central dogma of molecular biology. And it's really amazing. And if you are interested in the robots, and there is a longer version of that, okay? So this subject was covered in the earlier lectures, right? Okay, wonderful. Then what are the connections between the parts? As I said, there's no wiring, right? The connections could be the chemical reactions or associations, differed by whether a chemical bond is formed or not. Okay, so that's the connection. And today, I will start with introducing proteins, the workhorse of molecule, from 3 perspectives. First, biology. What do they do? Then, chemistry. What are they? Lastly, computing. How are they represented? So that's how we introduce proteins, okay? So let's, before we start, is there any questions or any clarification about vocabularies or anything? Any questions? Okay, so let's get started. Proteins, what do they do? Biology. This is really a workhorse molecule of life that is, active in every part of our cellular process, right? So I start with 2 examples, enzymes and antibodies. If you had a hamburger during lunch, it will be digested in hours, thanks to some kind of proteins called enzymes in your saliva, stomach, that can make this digestion much faster. So that's one kind of enzyme. There's another kind of that's one kind of protein. Another kind of protein is called antibody. That actually is part of our immune system to defend against the foreign invaders, like bacteria. Let's look at this one, enzymes, right? So enzymes can make some chemical reactions much faster. This chemical reaction is about breaking this green substrate into these two pieces, blue and red, called products. Okay, that's a chemical reaction. But this gray protein is the factory where this chemical reaction can occur much faster. And that is an enzyme. Okay? And here is an antibody, right? When a fresh wound is created, bacteria are invading, we have this kind of Y-shaped proteins called antibodies that are recognizing and neutralizing these pathogens. Okay, so we actually use antibodies a lot in drug discovery. So these are two kinds of proteins, what they do. Another kind, cellular messengers, right? So these are relaying the signals across cells and so on, right? Right now, at this moment, you are looking at the screen, you are processing the information, you are hearing what I'm saying, and you are thinking about something. What is the foundation of that? The foundation is the following. Photons are hitting your retina. And some proteins in your retina are sensing that and then relaying that chemical signal all the way to your brain. And then some proteins in your brain, called synapses, start firing electric current and shaping thoughts. So these are the cellular messengers. I give you two examples. In one example, this is a cell, right? Again, there is a nucleus and a cytoplasm, and then there's something outside of the cell. So there could be some environmental factors like steroid hormone that will be sensed and carried by a protein called steroid receptor. And then it will be carried into the nucleus and then triggers a lot of the downstream biological events. In another case, you don't have a receptor that moves things around, but it just sits in the membrane, okay? This is outside and this is inside the cell. So this G protein coupled receptor is both a sensor and an actuator. It senses the signals outside of the cell. And then it triggers the release of some proteins and the downstream effects inside the cell. So it's both a sensor and actuator. So in fact, both nuclear receptors and transmembrane receptors are common drug targets because proteins have so many functions in our body. But it's more than that. When you go to H-E-B and wonder why some brands of yogurts are more expensive, well, besides brands, maybe there's something about the protein content that makes the difference, right? Greek yogurt versus, the common ones, right? If you are building your muscles, right, you pay attention to your muscle milk or proteins because, again, there are these proteins called muscle proteins, like actins and myosins, that enables the muscular movement. And they are also active in pharmaceutical and other industry. For instance, in factories, they are in tanks to catalyze chemical reactions. Okay, so that's what proteins do. That's why we care about them. So then, if they are so amazing, what are they? What makes them so special? Well, very surprisingly, proteins are a very simple chemical structure. Despite the wonderful functions, it's a one-dimensional sequence of repeating subunits called amino acids. So it's a linear polymer, that's it. And what are the amino acids? Well, it basically has a carbon connected with 4 parts, right? So there's always a hydrogen, there's always an amino group, and there's always a carboxyl group. But then there's a 4th one, R group, or the side chain. That differentiates 20 types of standard amino acids. Okay, so the common parts of the amino group, carboxyl group, and central carbon is called the backbone. And then the distinguishing one is called the side chain. Okay? So it's exactly this distinguishing side chains that gives the different amino acids different personalities. And the different combinations of the amino acid type along the line gives proteins different functions. We don't fully understand that, but we try to. Okay. And as I said, there are 20 standard amino acids of different personalities shown in these green shadows. The rest are the common backbones: central carbon, amino group, carboxyl group, and hydrogen. And the green shadow is the side chain. They are very different. Some of them are hydrophobic. They like to hide from water and they like to congregate inside the core of a protein. Some are electrically charged, positively charged, or in this case, negatively charged and positively charged, okay? And some are just special. So because of their personality, right, they could be actually, linked together despite being very different. The amino acids can be linked together into a chain, right, through a peptide bond. So how does this happen? It involves a chemical reaction. So you see that the carboxyl group of the previous amino acid forms a chemical reaction of the amino group of the next amino acid. So then this OH group will leave, and this hydrogen will leave, and they together form a new bond and a new molecule called H2O, aka water. Whatever remains, called residues, will form a new bond between this carbon and this nitrogen, and that's our peptide bond. So that's how amino acids are linked together into a one-dimensional linear polymer, okay? But this one-dimensional sequence, right? Imagine that it's like a thread of colored beads. Each color is one amino acid type. So you could have all kinds of combinations. And this linear one-dimensional sequence, right, can be regarded as a text written in an alphabet of 20 letters. Well, each letter is a amino acid type, right? And this one-dimensional sequence actually has to fold into a three-dimensional structure before it can function, right? Like this three-dimensional structure in the cartoon representation. Well, it's not hard to understand if we think why geometry can give function. Let's think about why you can drink water from a mug. There is a handle and there is a bottom that holds water, and then there is an opening, right? But now let me change the shape of that mug. What if I remove the bottom of the mug and make the bottom significantly smaller than the opening? What do you end up having now? Is that still a mug? It's a funnel, exactly. So that's what I mean, that structure or geometry determines the function, okay, roughly speaking. So these proteins will interact with each other, in this case, hemoglobin that carries oxygen in our body consists of 2 copies of protein A and 2 copies of protein B interacting with each other. But proteins can interact with DNAs and RNAs and so on and function as well, okay? And so our study of protein structure, why do we care? Because it helps us understand how it functions, how it malfunctions in diseases, and how to design drugs to target proteins that malfunctions to treat diseases. So that's the implication, okay? So that's about proteins. And let's talk about the challenges. Challenge 1, we are accumulating a quickly growing catalog of the parts thanks to the high throughput sequencing technology. We know what protein sequences are in different species, right? Back in 2001, you need $100 million to sequence the genome. Now it's below $1,000. So there's exponential growth of sequence data, right? But for many of these parts, we don't know exactly what they do, partly because we don't know their structure, okay? So there's big data, but little knowledge. Let's visualize this gap between sequence and the structure. Over the years, even though I stopped at 2012, you see an exponential growth of the protein sequence data. In the meantime, the proteins whose functions are known or the structures are known remains much slower in growth. So this gap is widening, okay? So why? Part of the reason is that unlike the protein sequencing, which is high throughput, protein structure determination through X-ray, NMR, and cryo-EM can be very slow, okay? And not universally applicable. So because of that, computational prediction can fill this structure gap, where you start from the one-dimensional sequence and predict the three-dimensional structure. This is sometimes called the protein folding problem, but not exactly the case because, strictly speaking, protein folding cares more than the end of the folding process. It cares more than the structure. It actually cares about this whole movie, starting from the one-dimensional random coil and then eventually folding into a three-dimensional structure, okay? In fact, a lot of the neurodegenerative diseases are because of the protein folding pathway going wrong. And then, some diseases are caused, okay? That's a side note. So at this moment, I hope you have understood why is protein structure prediction needed. Okay. Is there a question? If you don't have a question for me, I have a question for you. Why is protein structure prediction needed?
I mean, like generally, I guess it's so that whenever there's issues like diseases and such, you have a basis for how to go off of your research, but also like you said, if you have more data, one, it's cheaper, but two, it's easier to understand future problems and current problems that people have. So
Wonderful, wonderful. I was going to say very shortly that structures are important, but not abundant. But you clearly explained why they're important. Okay, I really appreciate that. Very good, wonderful. Any questions or other answers? Okay, so now let me move on, okay? The second challenge. You're not only interested in predicting or knowing the functions of existing proteins. You may want to design proteins. In this case, anyone recognize what's the fella in the middle? Yes. Yes, it's the coronavirus with the spike proteins that are going to attack our cells. We'll see later on how they attack. But we can design these Y-shaped proteins called what? Antibodies that neutralize the spike proteins so that they can't attack us. We can design enzymes that can catalyze a reaction to degrade the plastics, PET, PE that we use a lot. We can design new species, rewiring the bacteria so that they can turn the dumps into electrons. That becomes a biofuel cell, right? And we can design proteins that can aggregate and form new materials for physical, chemical, electrical properties, right? So the challenge too is how do you design parts that have certain structures or function with certain parameters? This is not a big issue for resistors, capacitors, inductors. You all know by this point how to design a single layer coil with the desired inductance value, right? Some equations popping up, right? But if I ask you to design an enzyme that accelerates that plastic degradation by 1,000,000 times faster, well, you probably have to think about, well, what equation can I follow then, right? Not to mention that there are other proteins than enzymes, okay? Additional challenges are sometimes you want to introduce new parts, like drugs, that can repair these desired diseased systems. I talked about coronavirus disease, right? This is the protein I'm sorry, this is a virus with the spike protein that's going to actually attack our cell's membrane, right? And then it gets into our cells and then actually use the parts in our bodies and make the parts they need and keep attacking us inside. At every attack point, we can actually design some proteins or drugs to defend from the virus, like in this case. Before they get into the cells, let's introduce antibodies, right, that can wrap or neutralize the spike protein. In fact, they are one of the earliest authorized medicine for SARS-CoV-2, okay? And to solve this challenge, you have to solve the inverse problem, starting with the desired structure or even desired function, and then come up with the one-dimensional sequence that defines a protein. So that's the inverse problem, right? So you have prediction and design or problems that are inverse of each other. And I actually talked about this subject ever since 2017, the first time this course was offered. And it's a coincidence that 2 years ago, Nobel Prize in Chemistry was awarded in the subject of exactly these two topics, structure prediction and protein design, okay? I think John Jumper probably received the Nobel Prize only 10 years after PhD. And Demis Hassabis, keep in mind this name, because his name will appear again a couple of times in the next lecture. You will figure out actually what he actually started with. And so that's about the background of proteins, okay? So now we're heading into these two challenges. We focus on the first challenge, protein structure prediction, okay? So the question is the following: how do we figure out a shape of each part so that we can understand how it functions, okay? So the input is a sequence and output is a three-dimensional structure. And so now I need some input from you, right? So could you use your cell phone and scan this QR code, or just go to slido.com and put in this number. So you are a computational scientist, good at signal processing and everything, algorithms, right? I'm a protein scientist who's interested in protein structure prediction problem. We need to work together for this interdisciplinary challenge. So the question is, what information or questions do you want to answer from me so that you can solve this protein structure prediction problem? There are apparently two steps you have to achieve. You first have to formulate this problem mathematically or computationally, right? We're now doing experiments. Secondly, you have to approach or solve this problem with an algorithm, okay? So the question is, what information do you need to know? You know that the definition is from input sequence to output structure. You know the significance as just explained. So what do you need to know? Ask me anything to help us solve this problem together. Okay, let's see whether we get some responses already. I got one response. Can I get more, please? Okay, in the meantime, if anyone wants to speak up, just speak up. What question do you want to ask? So that we can approach this protein structure prediction problem. Anyone? Okay. Let's hope this number grows faster. Anyone wants to speak up? You want to try? Ask me anything.
I'm afraid not, no.
No? Okay, no question? That means you can already solve the problem, right? That's good. That's the best collaborator I need, okay? Other questions? Yes.
Are there specific protein substructures that have like repeated probabilities of showing up?
Wonderful, right? So repeated probabilities. So basically you're asking for the pattern in the existing data already, right? Very good, very good. Other thoughts? Okay, in the meantime, let's see. We got a dozen. Okay, I like this number 12, right? Okay. How accurate are the predictions? Well, we have to work on that together. Algorithmically, we can break the protein structure into parts. Good, you are decomposing problems. Good. How many different protein structures are present? Very good, you're asking about what are the possibilities out there, right? How would mutation work in this problem? Whoa, okay, the second stage collaboration. How can we leverage AI to help simulate the predictions? Wonderful, I'm all on. Just tell me how. Have there been similar sequences already processed? Wonderful, you're looking for data already. I would use clustering machine learning. Wow, you already have an algorithm. Wonderful. What are the amino acids you need? Or what R groups could be useful to know what they do? How do we represent or formulate proteins in a way that maintains their structural identities and amino acid chains while making them understandable and easy to work with computationally? I think that's a lot of thoughts. You are looking at not just computation, right? And other understandable or explainability. Wonderful. Specific patterns in the data. Is there a large data set to start with at all? And is there any redundant information? What are the input and output? I hope everyone understands now the input is a one-dimensional sequence and output is a three-dimensional structure. Maybe the three-dimensional structure alone is not quite clear for the moment, but bear with me. We'll explain what the three-dimensional structure means. Right now, you can think of it as an atomic cloud, okay, where each atom has a position. Okay, wonderful. So if I break all these answers, right, summarize them, and try to position your thoughts, okay, and also the next time you are in this position, people ask me everything. I tell you, there's a smart way to ask a question. Okay? Smart way is ask, what is the principle and what is the data? Okay? In other words, you're asking, how does a sequence fold into a three-dimensional structure? Do we know any principle about it? In the meantime, do we have a lot of data of protein sequences that already experimentally determined or not? Right? The rest is on top of it. Okay? So there is a spectrum of formulation in this principle versus data. Okay? And that's how we formulate the problem, and that's how we solve the problem. Okay? Now let me try to answer your questions. What's the principle? So we know that, again, protein sequences are of 20 different amino acids of different personalities. I told you that some of them are hydrophobic. They like to congregate with each other and hide from water. That's actually the first force that collapsed a one-dimensional sequence into a three-dimensional structure. But then there are all kinds of interactions that you have learned in physics, okay, including Coulombic interactions, right? And these interactions actually bring this protein into a three-dimensional structure. And how do I quantify that principle, right? Back in 1973, Chris Antonsen did some experiments to establish a thermodynamic principle that says the three-dimensional structure of a protein is corresponding to the Gibbs free energy of the whole system being the lowest. Oh, that's wonderful. You are telling me that there is a function of structure. And the best structure is when that function is the smallest. Wow, I have a way to formulate and solve that problem already, isn't it? Isn't it? Okay. So that's the principle. Now let's talk about data, right? The data are protein sequences with known structures already, right? So we want to ask the following. Do similar sequences have similar structures? So that when I have a new protein sequence, I can borrow some known protein sequence structures if they are similar, right? So that's the question I'm asked. So what I will do is the following. I will compare pairs, and I will compare proteins in pairs I will ask, what is the sequence similarity? What is the structure similarity? Then I will plot that in this two-dimensional sequence versus structure similarity axis, and each dot corresponds to one protein pair. And I will try to pay attention what's the pattern in that two-dimensional distribution, okay? That's the distribution you were asking for, I think, okay? So that's the distribution. We have two axes. Horizontally, we have the sequence similarity from the right to the left increasing. Vertically, we have the structure similarity from the top to the bottom increasing. Therefore, the lower left means identical sequences have identical structures, trivially. The issue is, what about the other points? Okay, let's ignore the colors for now. If we try to understand this pattern, you notice that at every sequence similarity level, like we fix the x-axis, then we notice that there is a lower frontier, which is the highest level of structure similarity that can be achieved. So this frontier gives you a most optimistic view of the structure similarity for a given sequence similarity. And if you look at the trend, in general, it says that the more similar the sequences are, the more similar the structures are. So it's very trivial, isn't it? However, you cannot just look at the frontier, because there are all these kind of points. Okay? So what we noticed was that if the sequence similarity is above 30%, the distribution is very narrow band. So we have a very good confidence that at this level of sequence similarity, structures would be very, very similar. Not a lot of outliers, right? That's what we call the safe zone. In the meantime, in this area, when the similarity is below 20%, then there is a wild possibility that corresponding to the high structure similarity and the low structure similarity. So that's a midnight zone that I cannot utilize data. So in other words, by looking at the data, I can come up with different scenarios. In each scenario, I have one way to formulate and solve this problem. Does that make sense? Okay, so the safe zone is easy to start with. Okay? So now let's come back to the protein structure prediction, right? So the input is a sequence X and output is a structure Y. And in that case, we have a structure prediction problem. And you have two ways to solve that problem, depending on whether you're a principal guy or a data guy. But usually we have to do something in between. So we might be optimizing a structure Y as a function of a sequence X, so that it minimizes the function of structure where sequence X is a parameter. So that's what semicolon means. Okay, so we're minimizing this energy as a function of a structure, and the best structure is the structure you are trying to predict. Another way is, let's use machine learning to predict this f of x from x to y. That's what AlphaFold did. Or you can even make it probabilistic, predicting the conditional probability of the structure given the sequence. Okay, so in other words, we are all building upon data homology. The only difference is the way you detect and utilize that homology. Could be superficial, could be shallow, but can be deep, as in deep learning. That's the only difference, okay? For now, we have the superficial view that the homology lies in sequence identity above 30%, okay? So let's look at the classical formulation. So let me confirm. We started from 220, we end at 355. Is that right? 335, okay, 35. Okay, I'll let you go before that, no worries. So the classical formulation, the easy approach when data is abundant. So in this case, the safe zone says that the query sequence whose structure is to be predicted is so similar to a template sequence whose structure is known. We can just copy the template structure. Somehow, copy it. okay, in a smart way. So that's a safe zone, okay? So that's exactly what we do, okay? And the way you do it is, for instance, this is a query sequence, and this is a template sequence, which is a one-dimensional text of alphabets, amino acid alphabets. You align them, and you figure out that, oh, outside of the box, I can use these parts, but inside the box, I have to somehow do some surgery. So I use the template structure. I figured out that these parts can be used, and then I try to fix the rest. That's called homology modeling, okay? So there are two problems specifically in the safe zone. The first one is there are a lot of template sequences with known structures. How do you find the best template quickly, right? And this is a database search problem, which is solved as a heuristic optimization. That doesn't guarantee you always ending up with the best solution, but it's really fast, okay? So it's a heuristic. So that's not going to be covered today. Another problem is if the template sequence is known and we have a pair of query and template, how do we align them and figure out that some parts can be borrowed and some parts have to be creative? That's called a sequence alignment problem, okay? So in this case, I will try to define a computational problem and then present a need for an algorithm without introducing the exact algorithm, okay? So it's a computational problem, okay? The problem is the following. We have two sequences, red and blue, query and template. And we want to insert gaps, like these hyphens, whenever applicable, okay? It could be in the red, it could be in the blue, to make these new sequences of the same length, okay? Then we want to make sure that we have a lot of similarities. In this case, red A, red T, blue T, so we have like 7 identities, right? 7 position-wise alignment out of maybe 15, okay? So that's our goal, okay? We are adding the gaps, hyphens, in the red and blue sequences to make them of the same length with the maximum alignment. So that's the problem. Is the problem clear? Does that make sense? Okay. So the question is, why do we need algorithms? Before that, algorithms sound very, far from us, but they are just procedures. Okay, that gives you the solution accurately and fast, okay? So in this case, we can come up with a brute force algorithm that just enumerates all the possible sequence alignment, okay? The issue is that it's not fast enough, okay? Let's figure out this question. How many ways, how many sequence alignments are there, right? Because for the brute force algorithm, the worst case is the last possibility is the solution, right? You enumerate all the sequence alignments, you end up having the last one. So, but that's the time you need to take. So how many sequence alignments are there for the brute force algorithm in the worst case? Let's try to answer this question. Well, you don't know the answer right away, but I assure you, all of you have almost solved this problem already in Ethan 303. So we have 2 extreme cases. In the shortest case, the shorter sequence is entirely enclosed in the longer sequence. In the longest case, there's no overlap, gap to character. So the longest is of length m plus n, right? Length of the red and plus length of the blue. You probably don't know how many sequence alignments are there. I'm going to do one thing. I'm going to transform each sequence alignment into a string, like the following. If this is an alignment, I'm going to go from red A, blue gap, but I ignore gap. Red G, blue gap, ignore. So I have A, G, A, G, A in red, and then blue A, red T, red C, red A, blue A. So that alignment becomes a sequence or string, right? How many positions are there in total? M plus n. There are m red and N blue, right? You cannot change the order within the red or within the blue, but you can, interdisperse them, right? Have you solved this Ethan 303 question? Imagine that there are M red marbles and M blue marbles. You cannot change the order within them, but you can, arrange them in a one-dimensional sequence. The question is, how many ways can you arrange their order? Does that ring a bell? Did you ever wonder, why did I have to solve this kind of problem? That's a prototype problem to motivate a lot of scientific discovery. Okay, now the question is, how many sequence alignments are there in total? Can anyone help me? This involves how to count without counting. In Ethan 303, there was permutation, combination, partition. What are we dealing with? Yes? Permutation. Okay, be careful. For permutation, you wonder about the order. But within red or within blue, I cannot change the order. If I change the order, I change the genetic information. Make sense? Okay. But I can, decide how to interdisperse them. Any suggestions? So in total, you have M plus N seeds. You are determining which ones are for the red and which ones are for the blue. All you need to do is to select M of the total seeds and reserve them for red. And then you don't care about the rest because among the red, there is an order already. There's no additional permutation. And for the rest of the seeds that are going to be taken by blue, right? So, or conversely, you select N seeds out of M plus M and reserve them for the blue, and the rest is for red. Therefore, we have M plus N choose M total number of alignments, as you all know after learning Ethan 303, right? And that's the worst case, okay? The issue is that is a very large number. If M is only 100, by the way, it's a very small protein. And that combination is 10 to 59. As a comparison, the universe age is 10 to 17. If you try to enumerate all the alignments from the Big Bang, think about whether you can finish that by now. So in that case, you need a better algorithm. It's called dynamic programming. You can reduce that 10 to 59 down to 10 to 4, M times N, where M and N are 10 to 2. So it's from 10 to 59 to 10 to 4, right? So you don't need to run the brute force algorithm to know that it's going to be slow, and you can design a better algorithm. Okay, that's the data extreme of the safe zone. Okay, we just solved a sequence alignment problem as optimization. And then if we go all the way to the other extreme where data homology carries very little information, right? When the sequence identity is below 20%, we can only rely on the principal now. So we have to find a optimal of this energy function as a function of structure Y, and we find the optimal Y that can minimize the energy function. Okay, so that is an optimization problem, right? Where you have a function, I changed the E to delta G, I apologize for that. So there's a function E of Y, structure Y, and you have a different kind of search space, and you have to find a search algorithm. So it's a search algorithm that we are looking for. Let me explain what we are facing. So the search space are the structures like the atomic coordinates x, y, z for each atom. Or you can represent them as angles, dihedrals, and so on. Doesn't matter. And what are the functions? Right? This involves forces and energy. Okay, like Yoda says. What energy you're talking about? Coulombic electrostatics, every one of you know, and all the other kind of, good old memories from the freshman year, right? And that gives you a challenge because you end up, apologize for a typo here, across that 74. You are in the high dimensional space. There might be thousands of atoms, each times 3 Cartesian coordinates. You are searching in that space. But this function is so rugged, noisy, okay? So it's like, you don't really have a map. You are searching in a thousands of dimensional space on a rugged terrain without a map. And you're trying to find the lowest point. All you know is at the next step, how high or high low I end up with, because I don't have the global map. You cannot afford calculating the function's value at every point. We simply don't have that map, okay? So how do you design an algorithm? That's what I mean. You don't need a lot of things to design an algorithm, because algorithm is what we tell computers do, and we tell them what to do very often from our own intuition. So let's say this is a high-dimensional terrain, right? Let's say you start from here. Then each time you go one step along some direction, and sometimes you end up being lower or better in energy. Sometimes you end up being higher or worse in energy. So my algorithm is the following. Every time I introduce a random perturbation going one step, if the proposed landing point is lower or better, good, I'm going to accept it all the time. That's going to be the starting point for the next step. But if it ends up being something worse, I don't reject it, but I accept it with a probability, okay? The more compromise, the less likely I'm going to accept it. My question is the following. Imagine that you are from the peak of the mountain and going back to the base camp somewhere at the foot of the mountain. So that's the minimization problem. You have to find a search problem. You don't have a map anymore, and it's dark. All you know is at each step, am I lower in the previous step or not? That's your search algorithm. So the question is, why don't I just accept a better or lower landing point every time? Why do I ever accept a worse, higher landing point in any step? Can I just always only accept a better landing point in each step? Yes? No? Why? Can I only accept the next landing point if it's better or lower? Any thoughts? There are local minima and maxima. Can we use the language of these terrain, mountains? By the way, you have the exact answer. I really appreciate it. Can we change that local minima to something about the mountains? Wonderful. So there might be a valley you end up being with. And every direction, any step is going to be something worse. If you don't accept a temporary setback, you will be stuck there. This is not protein folding or search algorithm. This is life, right? Okay, so then if I write algorithm in math, right, let me start with a x0, and then I perturb this into an x prime, and then I see whether it has a better energy or not. If it's not, I'm going to accept it with a probability. If this compromise is small, good. I accept it with a high probability. If its compromise is high, this is a low probability, okay? Don't tell me that I have climbed Mount Everest before I can reach the base camp, okay? But if you tell me it's just a small hill, I will do that. So that's algorithm. If it's something in between, what do you do? Well, we'll talk about it in the other lectures, okay? So now let's summarize, right? For the protein structure prediction, as I said, there are actually games that you can play to solve scientific problems, and you can be the authors of these papers. You can discover algorithms, actually, by playing games, okay? And you can find new protein structures by playing games. And there is a competition called a CASP where people participate and predict protein structures. And around 2018, echoing artificial intelligence, something happened. We'll explain what happened. And now let's ask any questions at this moment. How was protein structure prediction formulated and solved as computational problem? I hope that's getting clear, right? So now let's move on to what about the deep learning? Just a few words, right? So in the case of deep learning, right, what you're doing is like pushing to the extreme of the data. You're just using a bunch of X and Y pairs from the data, and then you try to learn a neural network that parameterizes the conditional distribution Y given X or structure given sequence. That's it, okay? So coming back to proteins, you can represent them as data. 1D sequence, 2D, 3D image or graph. They are very easily handled by modern AI or deep learning models like natural language processing, computer vision, graph learning, and so on. So you can learn them as if they are data. Back to CASP, around 2020, right? And during the competition, the organizers were amazed at one player or team whose performance is just way above everyone else. And organizers thought maybe someone is cheating. And it turned out it's not cheating, it's a revolution happened by an algorithm or software called AlphaFold 2, right? It trains on 170,000 structures on a neural network model using 16 TPU for a few weeks, okay? So this is tensor processing units. We'll explain more in the next lecture. The protein structure prediction that year and becomes the method of the year. And fast forward 3 more years, these 2 gentlemen behind AlphaFold won the Nobel Prize for protein structure prediction. And then there was AlphaFold 3 in 2024 and so on. So in the next lecture, we'll explain in more details how AlphaFold made it. So I hope at this point we are ready to talk about the last question I want to solve or challenge. That is a protein design problem, okay? So this is the inverse problem. So the inverse problem actually starts with a protein structure or function and end up having a sequence. So the computational goal, right, is inverse. How do I solve it? Well, why do I solve an energy minimization problem or something? Right? That for the given structure, I find the best sequence. I swapped X and Y. I need to figure out what's that utility function, okay, if you're a principal guy. Or I learned that f of x or f of inverse of x or p of x given y instead of p of y given x. Okay, so that's what we do. But how do you solve that exactly? First, the classical way. As I said, design is inverse to prediction. So the classical way of design approach is very similar to the classical way of prediction. Okay, so well, that's inverse. You want to start with a structure and find a sequence. What principle can I use? Energy minimum principle. Okay, so I can find the best sequence that minimizes that energy. But this problem computationally is challenging. I'll explain to you why, like that sequence alignment, okay? So the search space now not only include the atomic positions, which can be discretized by these sidechain rotamers, but also include the choice of the amino acids at each position because sequence is not fixed, right? So let's try to figure out how many how many solutions we have to go through until reaching the optimal one in the worst case. Let's say a brute force algorithm has n positions, okay, like 100 amino acids on this protein. At each position, we have 20 choices of amino acids, right? Because we are designing the protein sequence. At each position for each amino acid, the structure has k choices. So now at each position, you have 20 times k structure sequence combination choices. And there are n of them together. So by the basic rule of counting, you have 20 times k multiplied to itself for n times. Again, that is a huge number. Okay, I think it's way more than the number of planets in the universe. So in that case, you have smarter algorithms I will not venture into, but there's a huge success with protein design to catalyze reactions, and sometimes even for something that's never seen in nature. So in 2017, I made this slide when Francis Arnold designed an enzyme that make a bond between carbon and silicon. Nothing ever seen before in nature. Carbon, humans or Earth's lives are based on carbon. What lives are based on silicon? Robots. Then what does a bond between a carbon and a silicon may tell you? So some reporters say that, oh, look for the silicon-based alien life or iRobot. Anyway, that's a joke. But the year later, Francis Arnold was on the way to Texas A&M to give a talk, and he was woken up in the Dallas airport by a call from Sweden. And she won the Nobel Prize that year for protein design. And I really like this quote. It resonated with a lot of people. I want to read that. I always wanted to be a protein engineer. Proteins are marvelous molecular machines, tremendously complex, but responsible for all the functions of life. I wanted to be an engineer of the biological world. I hope this resonated with you as well. This protein design problem can be solved by classical approach and can be solved by deep learning as well. As I said, protein sequence structure can be regarded as text and image data. Around year 2014, thanks to a technology called Generative Adversarial Network, or GAN, a new word appeared called deepfake. Now we don't say that anymore. But the point is, you no longer recognize real images. You actually fake image, or let me call it synthesized images, or even videos. Okay? And the technologies are much better right now. Right? Let's look at these products from OpenAI from 2022 to 2024. You can use a text to generate an image, or you can use text, image, and chat. Right? This is GPT-4. Right? ChatGPT was GPT 3.5, and we now have 5.2. And now you can create videos by using text. Right? Just a few days ago, right, there was a new release from Google. You can create your own visual world and play game in it using text prompt. Okay? That means we can do something similar for proteins as well. You can use the same technology like GAN I was talking about, generative adversarial networks, then generate protein sequence and structures. Okay? And newest technology, if you use Midjourney or Stable Diffusion that create images, they are using a new technology called diffusion. So in the forward process, you crop the data to noise using a fixed schedule. In the reverse direction, you learn a neural network that starts from noise and then generate an image. So that's called a diffusion process model. And that can be used to design proteins as well. And again, David Baker won the Nobel Prize for protein design for his contributions in both the classical and the AI algorithms, okay? So at this moment, I think I'm close to the end. I just talked about proteins from 3 perspectives and why there is a need of structure prediction, right? And I also talked about how this structure prediction and design problem can be regarded as forward and inverse problems. How should we formulate them into a computational problem? And how should we approach them through algorithms? There was a clever way to think about the principle versus data, right? And maybe you find the sweet spot. And I have 3 takeaways for you today, if nothing else makes sense. First, protein structure bridges sequence and function, as she just explained at the beginning. But there's a gap of protein structure data, so we need these protein prediction methods that start from the sequence and predict the structure. In the meantime, we want to design new proteins, start with the structure Y and design the sequence X. And this can be done in optimization or machine learning, and AI really accelerated both. In the next lecture, we'll talk about not just drug discovery, but also the implication in healthcare. And this is the structure of the next lecture. I will talk about AI breakthroughs first, then show how they ripple into healthcare. Then I focus on the drug discovery before giving you the breadth of the impact in healthcare. Okay? At the end, I do want to give you some resources. How many of you have heard about this portal called Launch? Short for Learning, Academic Excellence, Undergraduate Research, National. Very good, you are utilizing it. So there are a lot of opportunities there for you to be aware of. The Student Research Week is in March, okay? I have 4 URS students who are presenting. There are many more students presenting their work then. You just need to register by February 24. Usually there are 2 workshops. One was gone, but the other one is in February, telling or teaching you how to give a talk, how to prepare slides, okay? But more importantly, you will get inspirations from the fellow researchers, okay? If you are interested in a longer time commitment in the summer, full-time, 40 hours a week, with salary and accommodation allowance, you can apply by February 16. You need just 2 letters, one from an instructor, another one from a faculty member who might be interested in working with you. And that's my point to the end. Always feel free to check with faculty members, okay? We may not always be able to respond, but focus on the responses you get and focus on the opportunities. We have a new program called ECE, Junior Research Scholars. Has anyone heard about it? Very good. So again, there are different areas that you might want to look into that connects you with industry. Of course, there is a university level undergraduate research scholar, right? At the end, you have a thesis and have some kind of ribbons during the graduation ceremony. And there are engineering opportunities, Aggie Challenge, and all these directed studies and research courses. All you need to do is send an email. There was a teenager who sent 200 inquiries nationally, 199 rejections until the last one. He worked with that Johns Hopkins University professor, and he invented a diagnosis tool for pancreatic cancer, okay? And fast forward, he gave a TED Talk, then now he's a master's student in electrical engineering at Stanford University. If you're curious about it, use Gemini to figure out who he is. At the end, I'd like to thank you for your attention. I'll be happy to answer any questions. I'll be staying around a little bit. Okay? All right. Thank you.